Hey, it's Miss Colangelo here. Today, because we can't do any in-class labs, we are going to do a video lab. Today, we will be doing the strawberry DNA extraction lab. Throughout the video, I'm going to be asking you guys some questions and then giving you a pause for you to formulate your own answer. Then I will go over the answer, so try to follow along. I will be going through the lab, and then afterwards we will be doing the analysis and conclusion questions. I would prefer that you guys would download either the PDF document that has all of the instructions on it or follow along on the PowerPoint that I also left. Both of those are on eCampus. If you do have all of these lab supplies and you want to follow along at home, please do. So let's start with some background information. All living things have DNA. Whether it's a human, a rat, a fish, bacteria, or even a strawberry. But what is DNA? Everything that happens inside of a living thing is controlled by DNA, just like a computer code. The computer programs can't run without a computer code. Anytime it's broken, the computer doesn't function properly, correct? So our DNA tells our cells how to run. It tells them what to do, when to start, when to stop. But where in the cell is DNA even located? DNA is stored in the nucleus of eukaryotic cells. This is the type of cell we'll be dealing with today. So what does DNA stand for? DNA stands for deoxyribonucleic acid. What does this mean for us though? It tells us that it is a nucleotide composed of a deoxyribose sugar molecule as the main structure, and then each one is attached to one of the four nitrogen bases: adenine, thymine, guanine, and cytosine. Each base is attached to that sugar and then is held together by a phosphate group. This acts as our backbone and strings together this into a long ladder-like structure. But speaking of the DNA structure, what is DNA shape? It is a double helix. Imagine that ladder but being twisted. Those long twists are then tightly wound and compacted into what structure? It's an X shape. Chromosomes. And that is how DNA is stored inside of the nucleus in the cell. So our goal today in that lab is to get the DNA out of the nucleus and unwind it into those long strands again. So what do we need for this? We need a strawberry, a plastic bag, a cheesecloth, or any other type of fine filter, a beaker or cup, 50 milliliters of dish soap, 90 milliliters of water, 2 teaspoons of salt, a stirring rod, and 20 milliliters of ethanol or isopropyl alcohol will work. So first thing we need to do is make our extraction buffer solution. To start, we need to mix our 50 milliliters of dish soap. With our 90 milliliters of water and 2 tablespoons of salt. Then give it a mix. So it should be a little bit thick. But why do we need this? Let's remember this structure of a cell and where DNA is located. The nucleus is in the middle of the cell, and to get to that, we need to get through the rest of the cell. The main obstacles are the membranes. the cell membrane and the nuclear membrane. To figure out how to break this, or break into this, we need to understand what it's made of. So, what is a cell membrane made of? A phospholipid bilayer. Let's focus on the part of the word that says lipid. Lipids are oils, fats, and waxes. The cell membrane is made out of long chains of fatty acids. When you're washing a greasy dish, what do you use? You use dish soap. Dish soap works by emulsifying fats. Emulsification is the breaking down of fatty acids. Dish soap causes a chemical reaction called saponification. This is where a fatty acid chain is broken into smaller pieces where the hydrophobic tails bind to the hydrophobic fats and the hydrophilic heads face towards the surrounding water. This means it forms an envelope around the broken pieces of fat and allows them to be washed away, just like when you wash your dishes. This tells you why we use soap, to break the membranes and allow them to be washed away. This also is why washing your hands kills the COVID virus. The membrane is made out of the same phospholipid bilayer and is broken down the same way with soap. This allows it to be washed away. Next, why do we use salt though? This is a bit more complicated and involves a lot of chemistry, but I will try to make myself understood. To start, remember that DNA is an acid. An acid carries a positive charge. Let's think more about DNA structure. The backbone in the phosphate group is what holds the DNA in a tightly wound chromosome. Salt helps break that and unravel it. But the next problem is the DNA that is still dissolved in water. DNA is normally stored in a mostly water-based substance, which is why it's dissolved in water. Water is what we call a polar molecule, and it carries a negative charge. Think about the poles of a magnet. If you hold a positive end to a negative end, they attract. And that is what happens here, and why it stays dissolved. And as long as it's dissolved, we can't see it. So we need to make them repel by adding salt to the water. Salt makes the water have a positive charge, and DNA has a positive charge. Think about those two magnets again. Two positive ends next to each other will repel. So now that DNA won't be dissolved, we need to make it precipitate or rain out. That's why we add the ethanol. So back to the experiment. Next, we're going to set our extraction buffer to the side, and we're going to grab our plastic bag. Put it up and toss your strawberry in. Mush. Get it into a very nice mushy mush. Yeah. So there's no big clumps. Once you think you have all those clumps kind of out, you're going to add some of the extraction buffer. I'm going to add about 10 to 15 milliliters of this. And then add it to your bag. And then mush again. And you're going to mix this and mush it around for about a minute to make sure that everything's very well mixed. Next, we're going to set up our filtration apparatus. So I'm going to take whatever cup you'll be kind of decanting into. Take a cheesecloth, and you can rubber band it, tie it, put it on a funnel, however you want to do this. Okay. What you're going to do now is run your filtration or your mixture through the filtration apparatus. So basically, dump your mixture into the cheesecloth over the cup. And now we're going to wait. But while we wait, I'm going to go through some fun facts with you. Fun facts about DNA, okay? So, if you're wondering why we use strawberries for this experiment, it's that strawberries have an unusually high amount of DNA in their cells for fruit. This makes it really easy to filter out. If you could stretch your DNA out into one long line, all of the DNA in all of your cells, it would stretch from the earth to the sun and back 600 times. Genes, or the active portion of your DNA, only make up about 3% of your total DNA. All humans are about 99.9% alike DNA-wise. We share about 60% of our DNA with strawberries and fruit flies, 50% with bananas, 98.7% with chimpanzees, 84% with dogs, and 90% with cats. If you could type 600 words per minute, 8 hours a day, it would take you about 50 years to type the human genome. DNA is incredibly fragile. Thousands of times a day, something happens that can cause an error. Errors can be caused by anything from problems in cell division to UV light. Many of these are repaired, but many are not, meaning that even you have mutations in your cells. Often they do nothing, some are helpful, and some are harmful and cause diseases. But technology called CRISPR, or clustered regularly interspaced short palindromic repeats, allow us to edit the genome and could one day help us cure genetic diseases like cancer, Alzheimer, and cystic fibrosis. Often Watson and Crick are credited with discovering the structure of DNA, but it was actually Rosalind Franklin. She was the first person to discover the structure of DNA. She did this through X-ray crystallography. This is where you diffract X-rays from a crystal, which show you the molecular structure in a three-dimensional image that we can actually see. So, back to our experiment. We've got a little bit going in here. I'm going to try to squeeze the rest of the little juice out. Okay. Next, we're left with this solution, or, mixture of strawberry. So next we're going to add about 20 milliliters of ethanol. We're going to, so this is our 20 milliliters. We're going to slowly add this. And then mix. You should start to see these like clumps form. Now, what I'm lifting and pulling out of here, that is actually all of the DNA. And you can see how it kind of separated and left behind like the clear-ish. So that is DNA. I'm going to try to move you guys in just a little bit closer so you can see more about the structure of it. Moving on. Make some observations about what you see right now. Do you guys still remember what this looks like and what you think? Let me know if you think this is, or if this is what you expected to come out of here, if this is what you expected the DNA to look like, okay? Let me know if you do this on your own, what your outcomes are too. Okay. So, moving on to our conclusion and analysis. I want you guys to follow along on the screen. So, the first one. It is important that you understand the steps in the extraction procedure and why each step is necessary. Each step in the procedure aided in isolating the DNA from other cellular materials. Match the procedure with its function. So, A, filter strawberry slurry through the cheesecloth. B, mush strawberry with salty, soapy solution. C, the initial smashing and grinding of the strawberry. And D, the addition of ethanol to filtered extract. Try to match these with the function on the side. So, the first one, to precipitate DNA solution. This matches up with D, the addition of ethanol to the filtered extraction. Second one, separate components of the cell. This matches up with the procedure part A, filter strawberry slurry through the cheesecloth. Next, to break open the cells, that matches up with C, the initial smashing and grinding of strawberry. And then the last one, break up proteins and dissolve membranes. That matches with B, mush strawberry with the salty soapy solution. Number 2, what did the DNA look like? Relate what you know about the chemical structure of DNA to what you observed today. Well, it was a long white strand, kind of clumpy. DNA would have been originally coiled up into chromosomes, and we broke up those binding proteins that keep them coiled together and expand them into long strands. So DNA, you know, is these long wispy strands, and you should be able to see that there are some of those wispy strands still. It clumped together into a mass because it is, you know, tiny and fragile, but you should be able to see that there's a bunch of little strands of it, okay? 3, explain what happened in the final step when you added ethanol to your strawberry extract. Hint, DNA is soluble in water, but not in ethanol. Because DNA is not soluble in ethanol, the DNA rains or precipitates out of the solution. But DNA is less dense, so it floats. Number 4, a person cannot see a single cotton thread 100 feet away, but if you wound thousands of thread together into a rope, it would be visible much further away. Is this statement analogous to our DNA extraction? Explain. Yes, it's very analogous to it. A single strand of DNA would not be visible, but here the DNA is clumped up and thousands of these strands are attached together, which makes it visible to the naked eye. 5. Why is it important for scientists to be able to remove DNA from an organism? List 2 reasons. There are quite a few. First one, genetic testing to figure out if someone has genetic markers or could have a possible disease, markers for that disease. Genetic modification, like we talked about in CRISPR. With CRISPR, you can actually take it out and change it. That's how they make insulin. Crime scene testing to find out the whodunit. You can find out your ancestry, and that's needed. You need to get the DNA out of the cell. And then research testing. Lastly, is there DNA in your food? And how do you know? Yes, all living things have DNA, or genetic material. Plants, animals, fungi are all eukaryotic cells, which means that it is all stored in the nucleus. Bacteria are prokaryotic cells, which means they are stored in the cytoplasm near the center. So, hopefully you guys found this a little bit interesting and fun. Follow along. I hope you enjoyed it and it wasn't too confusing. Have a good day. Stay safe. Stay healthy.